Assalamu alaikum. Today I will discuss about pharmacogenetics. This is an important branch in pharmacy and it is discussed in biopharmaceutics. Whenever we studied biopharmaceutics, the important term is pharmacogenetics. The pharmacogenetics is the study of influence of a gene on therapeutic and adverse effect of drugs. It is also defined as the study of inherited variation in drug metabolizing enzymes and drug response. Pharmacogenetics can be derived from two different words. One is pharmacon and another one is geneticos. Pharmacon means drug and geneticos means origin. That means pharmacogenetics deals with the origin or generative nature of drug. Primaquine induced hemolysis by in glucose 6-phosphate deficient patient was the first discovery of pharmacogenetics. The term pharmacogenetics was coined by Vosel in 1959. Currently, there are 120 drugs including voriconazole, warfarin, carbamazepine, cetuximab, etc., whose labeling includes pharmacogenetic discoveries. The FDA, that is known as Food and Drug Administration, publishes a guideline document to facilitate the use of pharmacogenomic discoveries in drug development. Pharmacogenetics, the division of pharmacogenetics is two types. One is drug disposition pharmacogenetics and another one is drug target pharmacogenetics. The pharmacokinetics of a drug can be altered by sequence variation in drug disposition pharmacogenetics, and the pharmacodynamic properties of drug can be changed by sequence variation in drug target gene. That means pharmacogenetics deals with both pharmacokinetic properties and pharmacodynamic properties of a drug. The drug disposition pharmacogenetics includes the distribution, absorption, distribution, metabolism, and elimination or excretion of drugs, that is known as ADME. The plasma concentration of the parent drug or its metabolites may be affected by a genetic polymorphism, altering the function of a protein that is involved in the disposition of a drug. For example, if a genetic polymorphism leads to the lower activity of a metabolizing enzyme, the plasma concentration of the parent drug may increase and plasma concentration of the metabolites may decrease. For example, warfarin and cytochrome P2C9 polymorphism. Another one is tamoxifen and cytochrome P2D6 polymorphism. To cytochrome P2C92 allelic gene results in a 30 to 40% reduction of enzymatic activity for S-warfarin metabolism, and cytochrome P2C9 cause complete loss of S-warfarin metabolism. Patient who carries cytochrome P2C9 or 2C93 or both causes higher serum concentration of S-warfarin. Study in Caucasian population causes significantly lower the doses of INR level when administering with warfarin in patient compared to patient carrying cytochrome P2C91 gene. Drug target pharmacogenetics cause pharmacologic effect of drugs excreted by modulating activities of enzyme or receptor. For example, vitamin K epoxide reductase complex subunit 1 gene, that is known as VKORC1, and warfarin response. Another one example is beta 1 adrenergic receptor gene polymorphism, that is known as ADRB1, and beta blocker response. Genetic polymorphism of drug target enzyme can be altered. VKWRC1 encode vitamin K epoxide reductase, which is inhibited by warfarin. This inhibition interferes with some coagulating factor 2, 7, 9, and 10 and anticoagulant protein C and S. There are two haplotypes, one is A and B. A haplotype cause the lower level of VKWRC1 mRNA expression compared to B haplotypes. Patients with A haplotype may produce a smaller amount of VKWRC1 compared to B haplotypes. These findings is true in Asian population. And when we discuss about ADRB1 and beta blocker response, there is SAR49 glycine and ARG389 glycine, two common SNPs for ADRB. It is hypothesized that patients carrying SAR49 or 389 would have greater reduction in blood pressure with beta blocker therapy. Several studies have found that hypertensive patients have both haplotypes and had the greatest reduction in blood pressure with oral metoprolol. Pharmacogenetics studies include candidate gene studies and genome-wide association studies. Genome-wide association studies, commonly known as GWAS studies. Both approaches can be used for gene disease and gene drug response association studies. These two studies include the total genetic variation in our body due to administration of drugs. The candidate gene studies are known as whenever a patient who have a better drug response, the candidate gene approach involves how frequently an allele or set of allele. Cytochrome P2C92 or 2C93 polymorphism had previously shown changes the function of 2C9 enzyme and were chosen to study the association of warfarin response. The candidate gene approach is useful tool to study a genetic association with drug response if there is a plausible link between gene and drug response. It is less expensive and requires shorter time. And genome-wide association studies have been conducted as case control, cohort, or family studies. It can be used to identify new biomarkers that could explain the underlying mechanism of adverse drug reaction. It includes genotyping large set of assemblies across the genome. The goal is to determine a particular allele or set of allele is more common in patients with certain disease or a better drug response. The role of pharmacogenetics in pharmaceutical industry includes studying drug metabolism activities, pharmacological effect, predicting genetically determined adverse reaction, and drug discovery and development, as well as planning for clinical trial. These are the role of pharmacogenetics in pharmaceutical industry. That means the role of pharmacogenetics includes both the pharmacokinetic properties and pharmacodynamic that we already included in previous slide. Pharmacogenetics has a threshold role in by pharmacists. The role of pharmacist may play a key role in pharmacokinetic discoveries. The role includes promoting the optimal use and timing of pharmacogenetic tests, interpreting clinical pharmacogenetic test results, educating other pharmacists and other healthcare professionals and patients and the public about the field of pharmacokinetics. Pharmacists can take a lead in application of pharmacogenetics in clinical practice since they are experts in pharmacokinetics and pharmacodynamics. Limitation of pharmacogenetics include the drug response is probably affected by multiple genes. Drug response might be predicted from a certain pattern of polymorphism rather than only a single polymorphism. Holding sensitive information on someone's genetic makeup raises question of privacy and security, as well as ethical dilemmas in disease prognosis and treatment of choice. That means there are some limitation that is associated with pharmacogenetics. This is pharmacokinetic properties or pharmacodynamic limitation or some kinds of ethical dilemma. There are some difference between pharmacogenetics and pharmacogenomics. Pharmacogenomics focuses on drug variability, pharmacogenetics focuses on patient variability. Pharmacogenomics includes many drugs and only one genome, whereas pharmacogenetics involves one drug in different patients with inherited gene variation. Pharmacogenomics predicts drug efficacy. On the other hand, pharmacogenetics predicts the drug toxicity level, and pharmacogenetics useful in patient healthcare and pharmacogenomics involved in drug selection. That means whenever we concluded our slide, pharmacogenetics is very important role in drug development and drug safety. Pharmacogenetics focuses on the effect of single gene in drug response. That is all from my discussion, and thank you all for listening my presentation.